So I'm going to start looking at the egg and the early embryo. I'm going to start off by orienting you. I work at Hammersmith Hospital, near Wormwood Scrubs Prison. My office looks into the prison yard, and it's a great view. And I've been working there since 1987, and I actually counted that up on my fingers and realised it was 25 years that I've been working there. I'm currently based in the Institute of Reproductive and Developmental Biology, which is this beautiful new building which was built round about the year 2000 with money raised by Robert Winston, something over £13 million, and it houses a lot of research groups who work on reproduction and development, and more recently on cancer. So I'm going to talk today about embryo development, about the very earliest stages of embryo development while the embryo is still in the oviduct or fallopian tube, and while it enters the uterus and implants. If you ask me about developmental biology after implantation, I get very, very hazy, so please don't. It was during these studies over 10, 15 years that it became increasingly apparent that the developmental potential and health of the embryo was dependent on what happened earlier, and encouraged me to start looking at the development of eggs in the ovary. So here we have a cartoon of the ovary containing eggs surrounded by somatic or nurse cells which develop and progress towards ovulation to produce that key cell in the body, the mature oocyte. So eggs, follicles, and early embryos are spherical. That is totally irrelevant to the lecture, but I just liked it because of the alliteration with the next 2 points, which is they're also scarce. There's not very many of them. And the worst thing is that they're small. They're very small. And just to give you an idea of how small, this is a picture I took on my iPhone, hence the high quality, of a ruler, 1 millimetre. This was actually Daniel's ruler. And Daniel, I'm really sorry about the fact that you had a GCSE exam today and you didn't have your ruler. That is my fault. 1 millimetre. I don't know if people can see, but there's a yellow spot here. That's the size of a mature egg, human egg, and the little tiny spot below is the size of a mature mouse egg. So you can see that we're dealing with structures that are really very small. So in order to investigate these structures, we use microscopes a lot. We have lots of microscopes and we have access to other microscopes, and I note that Tony McGee's just come in and he leads the film unit here at Imperial, and we use their microscopes, and thank you, Tony. And we need them to look at the development of embryos and to look at the histology of ovary. We also use molecular approaches, and the group have managed to optimize techniques for looking at small amounts of material, down to single or a few eggs. But another technique which I am going to talk about in some detail is we use mathematical models. And this is due mainly to the scarcity of the material. So we use models to inform our thinking and experimental planning. Now, I do want to say at this point, please don't worry about this. There is one equation in the whole lecture, so don't all leave now. So what I'm interested in is there are changes in cell shape in both embryos and early follicles. There's changes in how cells stick to each other, changes in how cells communicate with each other, and another thing is that I'm very interested in is the spatial organization within tissues, how different parts, different structures in a tissue actually communicate and influence each other. So I'm going to start with the beginning of my career in developmental biology, which was entirely accidental, I have to say. I actually wanted to be a policewoman. And I wanted to be a policewoman all through my teens. I watched The Sweeney avidly, and I wanted to be like John Thor. And fortunately, I don't look like him. And it was gently suggested when I was at university that maybe I could do something related to police work, perhaps forensic science. So I looked for forensic science jobs when I left university for a long while and couldn't find any. And an advert for a technician's job at the Medical Research Council came up, and I thought, well, I need a bit of money, so I'll apply for it. And I did, and I got the job working as a technician for the director of the MRC Experimental Embryology and Teratology Unit in Carshalton. And I thought that I'd stick it, you know, for a year or so. And this lecture focuses on, it's a tribute to people who've had a very powerful influence in my life and have changed the direction of my career. And these are two people who I owe a big thanks to. David Whittingham, who was the director that I worked for, and his research officer, Maureen Wood, who worked with him. And she taught me all the laboratory expertise that I know. And she got me interested in embryos and mouse embryos, and she gave me the most incredible and rigorous training. David Whittingham was famous for being the first person to freeze mouse embryos, transfer them, and get live-born young. And this is now a technique that is used worldwide for storing human embryos after IVF cycles. So this was the first animal experiment that showed that it was feasible. And you can see here more than 40% of the embryos in these pregnant mice gave rise to living full-term newborn mice. So it's very important work, and here he is celebrating, and he got the front cover of Science. Lucky man. So I'll talk a little bit here about preimplantation development. The egg is a very large cell. It's the largest cell in the human body, and initially it just cleaves into smaller and smaller cells. And then at the 8-cell stage in the mouse, something really quite magical happens. You can't see the individual cells anymore. And the cells increase how they stick to each other, the cells flatten, and this is a terribly important step developmentally, because the embryo now has an outside and an inside. And these outside cells are exposed to the surface, and the inside cells become what is termed the inner cell mass. And these inner cell mass cells will go on to form the fetus. And the outer cells, round here, go on to form the trophectoderm, and they are what will interact with the maternal reproductive tract, and they're involved in implantation. So during these very early few days, you've got some very exciting events happening with changes in cell shape, the assembly or building up of a fluid-filled cavity, and the formation of these two first lineages. So while I was at the MRC, I learned a number of techniques, one of which was how to activate embryos. Generally, an egg will be activated to develop by the fusion of a sperm with the plasma membrane. But it can activate if there are other stimuli. So, for example, if you gave an egg an electric shock or exposed it to dilute ethanol or changed the temperature a little bit, it can actually start developing and cleaving. And, you know, some reach the blastocyst stage. It is actually capable of implanting. Now, this is important because it demonstrates that the egg has most of what it needs to develop. It has everything there, bar the stimulus to start it cleaving. Now, that it does definitely need a sperm because these embryos are incapable of developing to full term. And you need the DNA from the sperm, which is marked in such a way that enables development to go all the way through. The other thing that I learned how to do, which was really, I loved doing this experiment, it's probably my favorite, was making chimeric mice. And to do this, you can take 8-cell embryos from white mice and from dark mice, and you can take their protective coat off, remove the zona pellucida, and you can place them next to each other, and they compact to form a single embryo. And they will develop into a blastocyst that's a mixture of white and black cells. You can transfer these blastocysts to a mouse, and you'll get some mice born from either the black embryo or the white embryo, but you will get some which are a mixture of the two. And you get these beautiful chimeric offspring, these stripy mice. And here are two of them. Here's a baby stripy mice, and here it is when it's older. And they're fertile and they're perfectly normal. But again, this is a very useful tool for looking at lineage development in mammals. So those were the kinds of techniques that I was learning. This, I think, is one of the most important men in my life, career-wise. He's a guy called Ray Wales from Perth, Australia. He was a very zingy Australian. He came with his wife to do a sabbatical at the MRC, and he'd got 3 kids about my age. And the best advice he gave me was dump the boyfriend, which I did, and do a PhD, which I eventually did, because at the time I was seconded to him to help him with his research. And we had tremendous fun together. This isn't, there we are. And what he taught me how to do was to make these little miniature incubation chambers, which are very tiny. You can see those are millimeters across the top. And we used to spend hours with super glue and glass test tubes making these little mini incubators for embryos. And these were for metabolic studies. And you can see that it had a resealable cap. And there's the little tube for incubating the embryo. There's a tube for gathering the CO2 that the embryo gives off. And you could culture embryos in here for 3 hours. And you had these little ports made out of cut-off syringe needles. So it was nothing if not imaginative. And you would thread in a glass pipette containing the embryo and put it very carefully in the tube, withdraw the pipette, pull out the syringe, and do these incubations. And this led to a joint paper that we did on the metabolism of glucose by human embryos. And this was working in collaboration with Ian Kraft, who was then at the Cromwell Hospital. And we were very interested in what embryos did in terms of their metabolism, because then it would help develop new and better culture media. Another lady who was very important was a lady, Audrey Muggleton-Harris, who taught me how to micromanipulate. And basically you've got a Petri dish with a drop of culture medium and this little embryo here, and you use these glass tools which are moved by these micromanipulators, and you can actually inject things or suck things out of the embryo. And she taught me how to inject cells into blastocysts, and it was very hard. It took me 6 weeks and I gave up. And I went to David Whittingham and I said, this was the kit that I had to use. It's fantastic. You can see here's the micromanipulator here. So you can literally, it's like a joystick, and you can do big movements with that. And it does tiny movements of the embryo down the microscope. This is a pipette puller designed by Audrey. I hope you can see the string and the spring and the filament for heating the glass. So you'd put a glass tube there, you'd heat it up with the filament, and it would go bing! And it was absolutely brilliant and very hit and miss. But anyway, I went to David and I said, I can't do this. I've got to give up. And he said, don't worry, Kate. He said, go to the bar. And in those days, MRC units had bars which sold alcohol. He said, have a G&T, come back after lunch and just mess about and play. So I went to the bar, I had a G&T, I came back, and 4 hours later, I'd injected about 100 blastocysts. So it was the best advice I'd ever been given, I think. Also working at the MRC was Alan Handyside. So here am I with very long hair, Alan, and this is my sister who's in the front row of the audience, Jo, and we're at a sports day at the MRC. And Alan was interested in embryonic stem cells. You can isolate these cells from the inner cell mass of the embryo. They're pluripotent. They have the potential to develop into any cell type, nerve, muscle, blood, whatever. And what's magic about them is if you inject them back into a blastocyst, they can take part in development. So they can be maintained indefinitely in vitro, unlike normal cells which will die after about 50 passages. And so Alan was collaborating with a chap from University of Edinburgh, Martin Hooper, who was already working on stem cells, and they had this idea of actually seeing if we could create a mouse model of a human disorder. And genetic diseases often result from loss of a key protein. So the disease that they decided to look at was one called Lesch Nyhan Syndrome, which is an inheritable and severe disorder. It mostly affects boys because it's X-linked, and men and boys only have one X chromosome. So if they have a mutation on that chromosome, it affects them very badly, whereas women have two X chromosomes, so one mutation can be counterbalanced by a healthy X. So it's caused by a lack of an enzyme called HPRT, and it's a very nasty disease. And one of the most horrific things is it causes this self-destructive behavior where the affected children sort of chew their fingers. So it was very nasty indeed. So what Alan did was created embryonic stem cells which lacked this key protein. They were isolated. I injected them into blastocysts and then transferred them into recipient mice so they could develop to term and produce chimeric pups, which we were successful in doing. And then these pups aren't just stripy on the surface, they're stripy all the way through. So everything's stripy. And they will have stripy gonads. I mean, they won't look stripy, but so they will have sperm in their testes, some of which are derived from the stem cells, and some of which are derived from the blastocyst. And it's the same in the ovaries as well. So these animals are very important. So you mate them with a normal, ordinary, wild-type mouse, and they will have offspring. And they will have some black offspring and some not black offspring. But if you're very, very lucky, you will get pale, in this case, offspring, which are derived from this HPRT deficient mouse line. So we've actually managed to get this mutation into the mice. And then you can breed the mice, and you can breed mice that are lacking HPRT. And I want you to remember that this was in the mid-1980s, so this hadn't been done much, and it was a very, very important experiment in the field, and it was published in Nature. And here is one of the first pups that was derived from these mutated embryonic stem cells. So this was very, very important because it now gave us a model to attempt this new concept of pre-implantation genetic diagnosis. And one of the great sadnesses of embarking on a pregnancy is that you hope that everything will be all right. And if both couples are suffering from a genetic disease, they've got a high chance that everything won't be all right. And at that time, in the late 1980s, the only choice for couples at risk of transmitting a genetic disease was chorionic villus biopsy or amniocentesis. This carries with it about a 1% risk of miscarriage, and it gives you that choice of a termination. And that is when, at a stage when you know you're pregnant and you feel that you're pregnant. And it's really a very ghastly choice to make. And the amazing thing about IVF is that you have embryos in a dish and they're accessible. And you can potentially remove cells from them, test the cells, and see if they're normal. So this was a very important idea. And by this time, Alan had left the MRC. I was quite heartbroken about that. And he defected to this lab in London that was run by Robert Winston and was going to go and work on developing pre-implantation diagnosis. I was still based at Carshalton at this stage, and the experiment that we all collaborated on was taking 8 cell embryos from these carrier females, taking off the zona, taking a cell off, testing the cell using a very sensitive enzyme assay, identifying embryos that we thought were affected and we thought were healthy, transferred them back, and checked to see whether we were correct. And we were correct. So this was a very, very important study confirming that it was possible to diagnose defects before implantation.
Can I ask a question?
I think it's probably, if we could discuss afterwards, that would be best.
Okay.
And so this was published in The Lancet in 1987. So the MRC was a fantastic place to work. It was a job for life, but technicians weren't allowed to do PhDs. And Ray had been banging on at me about, you've got to leave KD and do a PhD. So I went and joined Alan at the Royal Postgraduate Medical School and met Robert Winston, who interviewed me. He said he thought I looked very innocent, I recall. And I love this picture, actually, because what I like about this picture is the slightly quizzical, trying very hard not to look amused expression that you have on your face. So the plan when I moved was to do a PhD with Alan Handyside and work with both Alan and Robert on developing PGD for clinical use, and also, while I was at it, save the world and improve human embryo development in vitro. So this meant working with human embryos, and they can be donated for specific research projects, and it's done with the informed written consent from the couple and approval and a research license from the HFEA and ethics committee approval. So it's a very carefully regulated process, but there are many infertile couples who really do want to help research into their infertility and who do donate embryos. Human embryo development is very similar to mouse, so you have the cleavage, compaction, and blastocyst formation. It takes about 2 days longer. So in the mouse, development takes about 3 days, and in the human, 5 or 6 days to the blastocyst stage. So my first experiment was, how does biopsy affect human embryos? And so in this case, Alan would take cells off human embryos, and I looked at how they developed in vitro, in culture, how many developed to the blastocyst stage, and we found that if you take 1 or 2 cells off, these are control unmanipulated embryos, it didn't affect their ability significantly. If anything, it improved their ability to form blastocysts. And we also used a rather neat technique of counting the inner cell mass cells and the trophectoderm cells. The inner cell mass cells are in green, the trophectoderm in orange, to look at the effect of those at those all-important lineages. This is the inner cell mass, which would potentially go on to form the fetus. And you can see that removal of a single cell or two cells doesn't dramatically change the proportion or the number of cells in the inner cell mass. So this was a very important experiment, and it helped us move on to doing clinical studies. So this work was published in Human Reproduction, and we then moved on to do it for real. Here's a cell being taken off a human embryo. My job was to put the single cell in a tube to analyze it. And I have to say, it's probably the most stressful thing I've ever done in my life. So the first time I did it, the pipette was going, trying to get it into the tube. And you actually had to, it's on a microscope stage, so you had to see it go into the tube. But we did it, all of us. And I have to say that this was very much a team thing. Alan did the biopsy. I put them in the tube. Eleni Contogiani, who was a PhD student with us, did the molecular assessment, and Robert and his team looked after the patients, counselled them, worked them up for IVF, and also did the embryo transfer late at night when we finally got the answer. And I think, Robert, you were sometimes doing it after 9 o'clock later. Yeah. So it was a long day's work. And here are some of the first two babies, with Alan holding them, that were born successfully after this technique, and they'd been sexed because the parents were carrying a sex-linked disease. So it was tremendously exciting. It led to some quite interesting headlines, just what the doctor ordered. Okay. This one I wasn't quite so sure about. IVF team makes sex discovery. There's Alan, there's Eleni, who did the molecular, and there's me looking just like I do now. So that was very exciting. Meanwhile, there was a lot of work continuing in looking at the development of embryos, their metabolism, how cells differentiate, how cells stick to each other. So work carried on. And one of the things that we noted was that human embryos, unlike mouse embryos, they don't develop very well in vitro, and only about half of them make it through to the blastocyst stage. And the remaining 50% fall by the wayside at any of these stages. And we wanted to know why, and we wanted to know whether we could actually improve it with the environment. Were we doing something wrong with the culture system, or was it something fundamentally wrong with the egg itself? So I had two fantastic people working with me at the time on this problem. Anthony Leighton, who I believe now is working in Australia as a doctor. He belonged to the Territorial Army, and he liked parachuting. And I was always quite concerned as to whether he'd come in the next Monday in plaster or out of plaster. He was tremendous fun in the lab. And Fabienne de Vrecker from Belgium, who became a very good friend, also was working at the same time. And both of them did PhDs with me. And this slide is interesting. It's slightly depressing because it actually summarizes 10 years of work while we were fiddling around with the culture medium. So we'd keep an eye on the literature, we'd find out what worked in cow embryos and mouse embryos and hamster embryos, and we'd think, okay, we'll give that a go. So the first thing we did was reduce the glucose, and that improved things a bit. Then we removed it entirely, and that improved things, well, yeah, okay. Then we started adding amino acids, slight improvement, and then we started adding growth factors. As you can see, it wasn't stunning. And the conclusion that we came away with was that you have embryos which will grow, I mean, to be perfectly honest, they'd probably grow in distilled water with some Marvel added or something like that. They were just, they're what we would call resilient. And there were some embryos which just wouldn't develop. Even if they were in vivo, they wouldn't have developed. They could look beautiful, grade A, absolutely perfect, but they just wouldn't. And then we'd got a small group which, you know, could be affected by the culture medium. And this led us to think, in actual fact, we're not doing much good with the environment. It has to be the egg. So during this time, we'd been noticing also that another feature of embryos is that the cells fragment. This is a very common feature, is you get these little fragments. And a feature of apoptosis, or cell death, is fragmentation. So we thought, they're undergoing apoptosis. Now, apoptosis is generally a normal, healthy process. We need it. If we didn't have apoptosis, we wouldn't have fingers, because the formation of the clefts between the fingers is regulated apoptosis during development. It regulates hormone-dependent atrophy and hyperplasia, cell turnover in healthy tissues. You know, it's an important process, but too much of it can be a sign of problems. So we were using different techniques to look at the fragmentation of the nuclei and also the fragmentation of the DNA, another key marker. And this was work carried out by Sophie Spanos, who also did a PhD and who was a very talented PhD student. And over a decade, we kept careful records. So when we were counting cells in blastocysts, we recorded everything about them, how many live cells there were, how many dead cells. And I plotted a graph to see if it was related to cell numbers. And during this time, I got married. I married a mathematician called Jaroslav Stark. And he saw this graph and he said, "Oh, that's interesting. I like the look of that." He said, "I wonder if we can model it." And this wasn't the first time that we'd collaborated. He'd always been incredibly supportive of my research and had helped me with my stats and sums and things like that, and how to work out what 1 millimolar glucose was, and, you know, fundamentals like that. But this was the first big project that we'd done together. And what he did was he developed a virtual embryo, and it was using a model called a branching hypothesis, a branching model. And you can see it's obvious how it works. And the cells in this model, they can do three things. Here is the equation, the one equation in the whole lecture. So you can see it's very simple, don't panic. A cell can either die, it can do nothing, a bit like me, or it can divide. Now, I've had a number of rude comments from my family today about that picture, so we'll move quickly on. And this is the virtual embryo and what it can do. So the one cell can divide to 2, to 4, to 8, to 16, and so on. And it can go right up to the blastocyst stage. And this is all on a computer. When you're making a model, you've got to make it simple, otherwise you get lost. So the simplicity is the fact that the cells have got three choices. They can die, divide, or do nothing. They don't have any effect on each other. So if you've got a dead cell in one place, it doesn't matter to the cell next to it. It's not going to have an impact on it. The generations are synchronized. They have to be for the model, and they're not in real life. But, you know, hey, deal with it. And what you can adjust with this model is you can adjust the stage at which apoptosis starts and the rate at which it occurs. So, for example, you can have apoptosis starting really early at the 2-cell stage. So you can see here, one of the cells has died and the other cell carries on dividing, and you end up with a very small blastocyst with not very many cells. Or you can change the model so that it starts at the 16-cell stage quite late on, and it doesn't really have a big effect on the number of cells in the blastocyst. You can also change the probability of the cells dying. So you can have a lot of the cells dying here, or you can have very few of them dying. as in this model on the right. So these are all parameters that you can alter in this maths model. So here you've got few cells in this blastocyst and more cells in this blastocyst. And so we'd got 203 data points. So Yarostov ran this program 203 times for 203 virtual embryos. And he then plotted the results. So he would get the number of live and dead cells in that virtual embryo and the number of live and dead cells in that one, and so on, so that he had 203 data points from his embryos. And initially it wasn't very good. You want it to look like that graph there, and it doesn't. So I said, not impressed. Try again, tweak the model. And so this is basically what systems biology is all about, because, well, start anyway. You adjust your timing, you run it on the computer, you plot it, you compare it to the real data, you think, well, that didn't work very well, so you do it again and again and again. And during that time, you either get divorced or stop speaking to each other or whatever. But eventually, it worked. And the best fit of all, very interestingly, was if you had 3 different classes of eggs. If you had poor eggs, which didn't develop very well, eggs that had a modest death rate, and good quality eggs. And that was very, very unexpected and took us right back to that graph we had with the embryo culture data, that you've got differences in the origin of the embryo, differences in the eggs. And it all comes down, we concluded, to the egg. So I think we published this in PNAS and concluded that research on improving IVF success rates should focus on generating healthy gametes rather than on optimizing culture medium. So I moved gently, not overnight, my research to looking at eggs and the ovary, and started working with Stephen Franks. I want to put this rather curious photograph of him in a raincoat in context. I think Robert might remember what it is. It was in Finland. And there was a lady that we know, a Finnish lady, who decided that if we wanted to go to a conference in Finland, we had to make our own way there on a raft. So part of the trip was on this raft, and here's a picture of me looking slightly nervous and Robert in the background thinking it was all a big hoot. And we got very wet and finally arrived in Finland. Anyway, so I'm mostly using the mouse ovary for my research. Here is a juvenile mouse ovary, about 12 days old, and you can actually see the follicles in it, these little blobs in the ovary. We can culture these we can isolate the follicles, we can culture them, we can isolate eggs, we can do histology on them. So it's a very nice system to use, but these are still very small. They're still only 100 microns, 1/10 of a millimetre in diameter. So I haven't improved life for myself that much. And what we're interested in is how follicles start growing. Female mammals are born with all the eggs they'll ever have. And the vast majority of those eggs are very tiny, and they're resting, they're quiescent. And in the mouse, they look like these. They're very small with flat nurse granulosa cells around them. And they can start growing, the egg grows, the cells around them change shape, and this is something that we're very interested in at the moment, looking at how these cells stick to each other and how they change shape. And you get multiple layers of these granulosa cells, and I think you need more of these to support the growing egg. So it's a fascinating and very beautiful process that takes place, but it's one that we don't understand at all. Why is it important to understand it? Well, firstly, because it affects reproductive lifespan. So there's a big variation in how many eggs a woman is born with. And this is a sort of average curve of the decline. And when a woman runs out of eggs, she goes through the menopause. But if it goes too fast, you go through a premature menopause, and if it occurs too late, you can actually be so slow at producing growing eggs that there's a risk of having cycles where you don't ovulate at all. It's also an important stage because it's the start of massive egg growth. So this is to scale, and you can see how much the egg is growing in the mouse. And the egg is building up stores of proteins, messenger RNA, membrane, cytoplasm, organelles, mitochondria to make energy, and so on. So it's actually a very important phase. And what you're doing is building the large egg that will be the future embryo. So it's very important that these stages go right. It's not known what regulates this process. It's likely to be a local growth factor rather than hormones. We think that it's regulated by neighbouring follicles. So here we know, or there's increasing amount of evidence that the follicles themselves produce proteins, growth factors, which diffuse out and influence each other. So this is sort of network of growing follicles signaling to little follicles and little follicles signaling to bigger ones. And we don't know anything about these signals. We don't know if they're inhibitory or stimulatory. And so the question we asked was, what makes some follicles grow and not others? And this was very much a back of an envelope or serviette experiment. What we had noticed with our work, and others had noticed, we weren't the first ones by any stretch of the imagination to do this, but you usually get these tiny follicles in clusters. They're all around the outside, but they're often in clusters. And we were very, very interested in this spatial thing. And I said, well, do you think they're actually inhibiting each other? And we drew this little diagram because the follicles that start growing are usually sort of on the inner surface here towards the centre of the ovary, where there's no more follicles in the middle. And we thought maybe these are all signalling to each other, but these ones in the middle aren't getting so much signal because they're not surrounded. And so Yaroslav collaborated again with us in testing these hypotheses, and he supervised the mathematical aspects of this. And we found this fantastic program, which is like a James Bond film. It's called GraphClick, and it's got this magnifying glass. So you can take a histological section, put it into the program, and you can mark every single follicle. And you can plot its XY coordinates, so you know where all the follicles are in relation to each other. So this was very important and was carried out by a very talented postdoc who sadly was only with us for a year, Patricia da Silva Butkus. And if I tell you that the current lab is still using databases and forms designed by her, that'll give you an idea of how very good she was organisationally. And she collected a lot of these XY coordinates. And it's possible mathematically to calculate the distance between every single follicle in a section. So you get thousands of data points. I mean, if you tried to do this manually with a ruler, you'd die of boredom. And this was carried out by a very cool postdoc who, needless to say, was Italian, called Luca, a great asset to the lab. Mathematically, and he did all the programming and everything for this, and what he was able to do was separate follicles into groups. He could identify follicles that had no neighbours, they were all on their lonesome, they were completely isolated, follicles that had one other follicle close by, or two follicles, or three follicles. And we analysed this and we went back to our question, do these quiescent follicles inhibit each other? And the answer was a resounding yes. And this was so exciting. So basically, if you had a follicle and it had no primordial follicles nearby, it was all on its lonesome, it was likely, very, very likely to be a growing follicle. And as soon as there was one other follicle, one primordial follicle in the vicinity, the probability of it being a growing follicle halved. And the more primordial follicles you got in the neighbourhood, the stronger this effect was, and the less likely that follicle was to be a growing one. So it's very strong evidence that there was an inhibitory signal. And this was very, very exciting. And Luca drew this beautiful map. And so to actually model this, so focus on follicle 1 here, completely surrounded by other primordial follicles, all producing an inhibitor. And you can see it's completely bathed in inhibitor. It's not going to start growing. However, this follicle on the edge has only got one follicle nearby, and you can see that the amount of inhibitor is much, much less, and it's much more likely that this one could start growing. And here you've got a follicle all on its own, very little inhibitor, and it's likely, very likely to start growing. And this led us to the very interesting conclusion that this might explain why the number of eggs that a woman has, the number of follicles remaining, actually depletes faster as they get older, because the follicles become sparser and further apart from each other. So you don't have this strong inhibitory signal, so they start growing more and more. And it's likely to explain the classic curve you have of this precipitous decline after your mid-40s in the number of follicles. So now we knew where to start looking, and this is really informing our current research that we're doing now, which is very much focusing on the mouse ovary and what regulates the development. And we're searching for inhibitors using these natty techniques that we're developing. And the search continues. I now have 2 fantastic postdocs in the lab who are in the audience, Mark Fenwick and Jossie Mora. Mark isn't telling Jossie what to do. He's just learning from her. And this is the lovely culture lab that we have. Jossie does beautiful confocal images, looking at how cells stick to each other between the egg and the surrounding granulosa cells. And this is a technique that Mark absolutely hates, called in situ hybridization, which takes ages to do, but he is good at it when it works. And I'd like to finish by paying tribute to my late husband, who supported me so well throughout my career, really encouraged me to do a PhD, was a fantastic collaborator. We produced some very good work, and I hoped that it would continue. Sadly, it's unlikely to, with the systems biology and these maths modelling experiments. And he was also a very good husband, skiing companion, and father. So thank you, Yaroslav. And this lecture is a tribute to you. It's also a tribute to people who I think have really influenced my career. A big thank you to Steve, who's been a very good collaborator for more years than I care to remember. Ray, he was a surrogate father. I think I was a bit of a surrogate daughter when he came on sabbatical. We had a really fun time in the lab, and he was the person who said, go for it. And it's a big thank you to him. A big thank you to Malcolm for putting up with me for 10 years. He's actually looking at the new building as it's going up, but Steve said he's probably wearing the hard hat to keep the flack off him while he was head of department. And an enormous thank you to Robert for paying my salary for many years, being so supportive, making the odd pithy comment like, that looks like a worthy paper, Kate. Thank you, all of you. And I'd also like to end up by thanking all the people who've worked with me with these various studies, Anthony and Fabienne, people who worked on the cell death, the mathematical modelling, and the ovary studies, particularly Sharon, who I know is in the audience just behind there. Hi, Sharon. who ran our lab so beautifully for a decade before moving on to pastures new. So I will end there. Thank you very much.